rhododendron species is one of the rarest flowering plants in the world. Overall, the rhododendron species has been classified as threatened and endangered species. The Malaysian rhododendron of Peninsular Malaysia especially is considered as vulnerable to extinction. According to Sarkar, one of the threats faced by rhododendron species is habitat degradation in the protected area. This is caused by both natural and anthropogenic disturbances, which had a great influence on the natural forest resources. Additionally, the condition of the habitat of rhododendron, which is Cameron Highlands, also becomes a problem to the availability of this species. Cameron Highlands has been affected by the rising in temperature due to the climate change and global warming, which lead to habitat disturbances. The objective of the research study is to identify the type of rhododendron species, to determine the morphological structure of rhododendron species, to determine the type of pollinators involved for rhododendron species, and to determine the abundance of the rhododendron species, specifically at Mentigi Hill and Mount Irau, Cameron Highlands, Malaysia. The significant study of this research is to help students, researchers, community and the government. Firstly, the significant study toward the students to be able to make this study as the guideline and aid in doing their research proposal papers with more depth. Next, the significant study toward researchers is to make this study as references in having more new research about rhododendron species, especially regarding the species abundance and the pollinator agent that exists in Cameron Highlands. This wood flowering plant species has few discovery in the Peninsular Malaysia, thus the research on this particular plant species is highly encouraged. Other than that, the significant study toward the community is to expose the community in our country about the existence of this beautiful plant species, as well as to inform them that this endangered species need more attention. Last but not least, the significant study toward the government is to maintain the population of the Malaysian rhododendron species, where only 15 species are available throughout the nation. It is hoped that this study is able to further prove that rhododendron are worth investigating with more depth, as there are so much things that we did not know about this unique species. The study on the type of pollinator involved, number of species or species abundance, as well as the morphological studies on rhododendron species will help us in understanding more about the species. The study was conducted at Mentigi Hill and Mount Irau, Cameron Highlands, Pahang, Malaysia. The rhododendron that exists in Malaysia is known as Malaysian rhododendron. In Malaysia, specifically in the Peninsular Malaysia, there are exactly 15 species of Malaysian rhododendron, including the natural hybrid, endemic and non-endemic species. Plants utilize pollinating agent for the purpose of pollination. Pollinating agents are divided into two types, which are biotic and abiotic agents. The example of biotic agents are animal, insects, butterfly, birds and so on. While the example of abiotic agent is wind, water, temperature and so on. The pollinators plays important roles in determining the floral density of a certain species. This table here shows the characteristic of pollinated plants by 4 different pollinators agents, which are the animal, insect, wind, and water pollinators. Rhododendrons have several importance in many fields, such as medical field, industrial field, and horticultural field. Researchers have discovered that the blossoms of certain rhododendron species are useful in treating a wide range of illnesses, including those related to detoxification, inflammation, constipation, bronchitis, asthma, dysentery, diarrhea, fever, and heart disease. One of the apparatus that we use during data collection in Cameron Highlands is Haga altimeter. It is used to calculate the tree height, but we also use smart measurement app to get more precise data. Environmental meter was used to find the actual wind flow in Cameron Highlands. Meanwhile, the measuring tape was used in making a plot for the plot sampling method. A ruler of 30 cm long was used to measure the rhododendron leaf length. The other apparatus, such as rubber mallet, raffia ropes, and wooden stakes, were used in the plot making processes. The methods and techniques in the research includes plot sampling method, leaf recognition method, clustering method, as well as observation on the surrounding environment. These are the pictures that were taken during the data sampling and data collection activities in both Mentigi Hill and Mount Irau, Cameron Highlands, Malaysia. Types of species of rhododendron that we found in Mentigi Hill, Cameron Highlands are as the following. In Mentigi Hill, we also found a few types of insects which are potential pollinating agents for rhododendron species. There are 5 plots that were made in 5 different areas in Mentigi Hill. From the first table, we can see that the highest elevation is on plot 1 with 5,151 feet. The lowest elevation is on plot 5 with 4,961 feet. However, we observe the highest wind flow occurs in plot 1 and plot 5. The second table shows the species type and number of individuals that were found in each plot. There are a total of 9 rhododendron species found in Mentigi Hill. However, no species were found in Mount Irau, as we were unable to reach the peak of the mountain to collect the data. For data analysis, we used bar graph and scatter chart to show the pattern in both elevation and wind flows for 5 different plots. The pie chart shows the number of individuals per species in percentage form. It shows that Rhododendron boreavi has the highest percentage with 31% of overall species in Mentigi Hill. The other 2 graphs shows the differences of tree height and leaf length among the rhododendron species. Majority of the rhododendron species in Mentigi Hill, Cameron Highlands are native to other countries. Only some species are native to Malaysia. There are 3 species native to northern India, 2 species native to China and East Asia, and 2 species native to Indonesia, Philippines, and Papua New Guinea, and the remaining 2 species are native to Peninsular Malaysia. Rhododendron longiflorum can usually be found in Mount Rincang and Mount Tahan in Pahang, whereas Rhododendron jasminiflorum is found along the peninsula of Malaysia. Rhododendron is divided into 2 categories, which are lepidot and ellepidot. Lepidot is small leaf plant, however, ellepidot is large leaf plant. Mentigi Hill consists of many Viraya species or Malaysian rhododendrons such as Rhododendron jasminiflorum, Rhododendron longiflorum, and Rhododendron javanicum. The ellepidots found in Mentigi Hill are Rhododendron simpsi and Rhododendron johnstonanum. Noted that there are also some species that are categorized into ellepidot even though they have small leaf. Wind flows contribute to the increase in number of individuals for rhododendron species. The species with the highest number of individuals was recorded to appear in plot 1 and plot 5. This proved the existence of wind pollinators. Usually, the highest elevation has the highest wind flow, but the geological structure can also contribute to high wind flow. For example, an open area has a big surface area which exposes them more to the wind. Based on the observation of the study area, we found that plot 5 is located in a windy and open area, meanwhile, plot 1 is located at the highest peak of Mentigi Hill. We also found the presence of insects around the study area. Insect species such as tachinid fly, Paracirpus hoverfly, weevil beetle, spider, and millipede were found in almost every plot, which determines the existence of insect pollinators in Mentigi Hill. Both plots experience the strongest wind, which contribute to the activity of insect pollinators to pollinate the flowers rapidly. Rhododendron boreavi and Rhododendron longiflorum in plot 1, as well as Rhododendron zoileri and Rhododendron simpsi in plot 5 has the highest percentage of number of individuals. Other rhododendron species that we obtained during the data sampling also has a significant number of individuals. This shows that most of rhododendron species in Mentigi Hill are either or both insect pollinated and wind pollinated plants. We conclude that a various type of rhododendron species exist in Mentigi Hill, Cameron Highlands, Malaysia. The flowers of this species occurs in various magnificent colors and shapes. They have a wide variation of tree height, Some species occurs in evergreen shrub and some species occurs in tall woody trees. Rhododendrons is distinguished into two groups, lepidodes and elapidodes. Lepidodes are small leaf species and have a minute scales underneath their leaves. Meanwhile, elapidodes are large leaf species and lack the minute scales. Malaysian rhododendron or Viraya species as well as the azaleas are found to live in a cool climate such as Mentigi Hill. A total of 9 species of rhododendron were found by implementing the plug sampling method. Wild rhododendron, especially the one living in a tropical forest such as Mentigi Hill and Mount Irau, are distributed mostly on high altitude area like the hilltop or the mountain peak. Wind flow as well as insects are the main factors of the distribution and abundance of the rhododendron species in Cameron Highlands. The number of individuals was calculated during the expedition in Mentigi Hill, and we determined that high altitude, presence of insect pollinators, and strong winds play important roles in the reproduction and survival of rhododendron in Cameron Highlands, Malaysia. Throughout the data sampling activity in Mentigi Hill and Mount Irau Cameron Highlands, we encountered a few limitations. Firstly, we lacked time to perform the data collection. A total of only 2 days were given to us to completely collect all the data regarding the rhododendron species. The second limitation that we face is the miscalculation of the season of rhododendron flowers to bloom. This species is supposed to fully bloom in spring season, which is between mid-March to mid-May. However, we had conducted our research in mid-May, which is a bit late. As a result, we were not able to observe the flowers of rhododendron species in Cameron Highlands. Thus, this research study only focused on the leaf morphological features of rhododendron species as well as the environment of the study area. As well as, all in all, we recommend that the research should expanded to a week at least to obtain maximum number of data collection.